Assalamu alaikum. In this lecture, I will be discussing about the chemotherapeutic agents. Chemotherapeutic agents, what are the different source or what are the different compounds which are used against the microorganisms, like we are having list of chemicals or drugs. We are having list of chemicals or drugs that are used against different types of microorganisms. These drugs or chemicals inhibit or destroy these organisms. As we are having many organisms, bacteria is there, fungus, virus, or parasite. So against these, we are having many chemicals, like we are having antibiotics, we are having antimetabolites, alkylating agents, or mitotic spindle inhibitors, and many more are there. So these compounds or chemicals are called accordingly. Like we are having antifungal, which are used against the fungi, antiviral against the viruses, antiprotozoal, or antibiotics in general we call them as, or anti-neoplastic. We call these chemotherapeutic agents also anti-neoplastic agents. They directly or indirectly are going to control or inhibit the growth and proliferation of cells. We are having many classes of these compounds. They are classified according to their mechanism of action, how these anti-therapeutic agents are acting. Like we are having alkylating agents, antimetabolites, or many others. And when these compounds or drugs or chemicals are used against treating any disease, we call that as a chemotherapy. So the chemotherapy is the treatment of disease by chemical compounds. Until the end of the 19th century, most drugs were derived either from minerals or from plants. Then the existence of Louis Pasteur came. He was able to isolate drugs. He knew how to use the drugs. It was Paul Ehrlich who did the greatest contribution to the science in case of chemotherapy. Paul Ehrlich was a scientist who was the one who did a great contribution to this chemotherapy. The discovery of penicillium by Sir Alexander Fleming in 1928 and its practical development by Sir Howard Floyer and Ernest Jane marketed another important advance in bacterial chemotherapy. Now the antibiotics. These are chemical substances which are produced in environment by different types of organisms. These organisms are having ability to kill or suppress other organisms. Like from these organisms, we are having to able to get penicillin or streptomycin. We call these as an antimicrobial agent. Now, what are the antimicrobial agents? These agents may be either naturally occurring, or we can synthesize, or we can take compound from one natural source, one from artificial source. So we call such agents as a semi-synthetic chemical substances. These antimicrobial agents are having ability to kill or suppress the growth of other organisms. Category of antibiotics. Like we are having two categories for antibiotics. One is bactericidal. Another is bacteriostatic. Now, bactericidal. These drugs kill target bacteria or pathogen. Means they are specific in their action. They are more effective and more preferred one. These bactericidal are they are directly targeted to the single type of bacteria or pathogen. When we are having infections, when we want to get the defense, when we want to increase our mechanism, if we are infected with something like meningitis or something, bone marrow, that time we need to that time when we need to eradicate these organisms, that time we are using bactericidal. जब हमें क्या है, हमें किसी एक पर्टिकुलर जगह पे क्या है, प्रॉब्लम है, इन्फेक्शन है, हमें उस जगह को रिमूव करना है वो इन्फेक्शन, उस टाइम पे हम लोग कौन सा ये लेते हैं, बैक्टीरिया, हम लोग कौन सा एंटीबायोटिक्स लेते हैं, बैक्टीरियोसिडल, बिकॉज दे आर direct targeting the bacteria or pathogen. We are having many examples which are bactericidal in nature, and the examples are fluoroquinolones, beta-lactams, or aminoglycosides. Aminoglycosides, beta-lactams, or fluoroquinolones are the examples of bactericidal antibiotic. Now the bacteriostatic. Now the another category of antibiotics is bacteriostatic. These drugs reversibly inhibit the growth of bacteria or pathogen. For these drugs, duration of therapy must be sufficient to allow cellular and humoral defense mechanism to eradicate the bacteria, खत्म करना है बैक्टीरिया को, as they allow the normal host defense to destroy the pathogen. Means ये naturally ये क्या करता है, मतलब allow करता है like cellular और humoral defense mechanism को eradicate करने में बैक्टीरिया को. Like we are examples for this is sulfonamides or chlorophenicol or chlorophenicol. Now the second class of chemotherapeutic agents are antimetabolites. Antimetabolites chemotherapy agents belong to the cell cycle specific drugs. These are the cell cycle specific drugs. They inhibit DNA replication or repair by mimicking normal cell compounds, or they act as S-phase specific. It means जो हमारे ये एंटीमेटाबोलाइट ड्रग्स है वो कहाँ पे एक्ट करता है, वो सेल साइकिल स्पेसिफिक ड्रग्स होते हैं. They will act on inhibit DNA replication बैक्टीरिया का, या तो वो रिपेयर मतलब कुछ ऐसा कंपाउंड उसमें डालते हैं जो नॉर्मल सेल के कंपाउंड से मिमिक करता है तो उसको स्टॉप कर देते हैं, या S-phase स्पेसिफिक होते हैं वो, या तो S-phase स्पेसिफिक होते हैं. The different cell cycle phases include G1, S, G2, and M phase. So these antimicrobial and antimetabolites target the S phase when DNA replication occurs, thus inhibiting DNA synthesis of many cells, of many bacteria, if it is infection is of bacterial or if tumor cells or cancer cells. Even in this antimetabolites, we are having pyrimidine inhibitors, we are having purine inhibitors. The example of the pyrimidine inhibitors are 5-fluorouracil or citrabine. And the example of purine inhibitors are 6-mercaptopurine or furabadine or furabadine. But they are having these antimetabolite chemotherapeutic agents are having one problem. The cell cycle specific chemotherapeutic drugs can't differentiate. They don't have ability to differentiate the healthy cells or the infectious cells. Means वो जाके सीधे अटैक कर देते हैं एस फेज में कहीं पर, चाहे नॉर्मल सेल हो या तो बैक्टीरियल सेल या कैंसर सेल या ट्यूमर सेल हो, जाके अटैक कर देता है. तो इसकी वजह से क्या होता है कि जो हमारी नॉर्मल सेल ग्रोथ है वो भी हैम्पर हो जाती है. Now the third class is the alkylating agents. These agents were the first drugs which were used as anti-cancer drugs, and in most commonly used agent in today's world. It means alkylating agents जो है वो हमारे एंटी कैंसर ड्रग भी यूज हम लोग उसको उसमें भी यूज करते हैं और आजकल के जमाने में ज्यादा चलता है. These alkylating agents act directly on DNA or causing cross linking of DNA strands, और either ये abnormal base pairs बनाता है or even they are having ability to break the DNA strands. In short, ये क्या करता है? They are preventing the cell from division. Alkylating agents used for most types of cancers. एक कैंसर के लिए काम नहीं होता, ये बहुत सारे कैंसर सेल्स के लिए काम करता है. And its greatest value is in like they are having ability to treat slow growing cancers. जब कैंसर की रेट क्या होती है, स्लो होती है, तब जाके ये बहुत efficiently act करता है. Alkylating agents are not as effective as on rapidly growing cells. अगर ये हमारी जो कैंसर सेल्स है वो ज्यादा फास्ट डिवीजन कर रही है तब हमारे ये alkylating agent उस पर काम नहीं करते. And the best example of alkylating agents are nitrogen esters, hydrazine, platinum-based agents, or oxophosphorins. Even it includes or chloroampicillin. The another class is mitotic spindle inhibitors. These are the drugs which are used to inhibit the mitosis or the cell division. What is their role? What these mitotic spindles do? We know that these drugs is going to disrupt the microtubules. Now, what are these microtubules? Which are the structure that pull the chromosome apart when there is cell division is occurring. These mitotic inhibitors are actually used in treating the cancer. These are used in cancer treatment because we know that cancer cells are rapidly growing, dividing and spreading easily. So there is continuous mitotic division. Then if there is continuous mitotic division, then these cancer cells are more sensitive to inhibition of mitosis than normal cells. Mitotic inhibitors are also used in cytogenetics. The best examples of mitotic spindle inhibitors are taxanes, vincanculides, vinblastine, vinorelbine. These are the examples of the mitotic spindle inhibitors. Vinblastine or vincanculides were the first natural products used for chemotherapy. These compounds and their analogs bind freely to the tubulin, which prevents tubulin aggregation and polymer stabilization. But these compounds is said that they are having significant side effects as well. Vincanculides are actually amines. These are plant products. Vinblastine is used to treat the leukemia, breast cancer, and testes cancer. Vinorelbine is used to treat breast cancer and non-small cell lung cancer. And the most commonly which we have heard about in day-to-day life is the colchicine. Colchicine is an alkaloid-derived compound. It inhibits mitosis by inhibiting microtubule polymerization. While colchicine is not used to treat cancer in humans, it is commonly used to treat acute attack of gout. So we are not using colchicine as a chemotherapeutic agent. In others, we are having enzymes, antibiotics, protease inhibitors, tyrosine kinase inhibitors, which act as a chemotherapeutic agents. PDP enzyme is one of the mostly commonly enzyme used as a chemotherapeutic agent. PDP enzyme. More organic compounds are used to inhibit the function of the protein, which act at the RNA level, so that if the compound is act at RNA level, there will be no formation of the proteins. We also have topoisomerase inhibitors. These are the drugs that affect the activity of two enzymes, topoisomerase 1 and topoisomerase 2. When the DNA double strand helix is unwound during the DNA replication, or it's unwound at transcription level also, the adjacent unopened DNA wings tighten. That is, if they are getting tight, it means supercoiling is occurring. Like opening the middle of the twisted rope, जो वो V shape बनाते हैं twisted rope जैसे, पीछे से Y shape आ जाता है, वहां पे क्या होता है, the opening of the middle twisted rope, the stress जो है वो कौन एड करता है वहां पे, वो स्ट्रेस टोपोआइसोमरेज एंजाइम. जब हम लोग डीएनए को अनवाइंड करते हैं फॉर रेप्लिकेशन और ट्रांसक्रिप्शन लेवल, वो जो स्ट्रेस आ जाता है बीच में, वो कौन करता है, बेयर, वो एड करता है. किससे होता है, वही टोपोआइसोमरेज एंजाइम से. दे प्रोड्यूस सिंगल और double stranded break into the DNA, reducing the tension of the DNA strand. This allows the normal unwinding of DNA to occur during the replication or the transcription. So inhibition of these topoisomerase 1 or 2 interfere in both these processes. जब जो ये हम लोग ड्रग टोपोआइसोमरेज इनहिबिटर्स यूज कर रहे हैं, उससे क्या हो रहा है, उसकी वजह से ब्रेक हो रहा है, तो नॉर्मल अनवाइंडिंग हो जा रही है, तो वो फिर रिवाइंड वापस से नहीं हो जाता वो डीएनए क्वालिटी. So this is how the topoisomerase inhibitors are used as a chemotherapeutic agent. Now the selection of antimicrobial agents. Now we have to decide which antimicrobial agent or which chemotherapeutic agent we have to use. It depends. Our target organism is either fungi, bacteria, or protozoa, or virus. As per our organism, we have to take that chemotherapeutic agent. Even if we talk about the drug which we are selecting, it also is related to many factors. Like if we talk about the type of the activity, which type of activity we want to perform, or how the drug is interacting with our microbe, how this drug is going to have impact on the lifestyle or the growth and development of the microbe, how we are going to administer this chemotherapeutic agent into our body, what is the related toxicity of this drug which we are administering in our body, and finally the cost of the therapy, the agents which we are using as a chemotherapeutic agent, what will be its final cost, what will be its final. All these parameters has to be selected before we are going to use these antimicrobial drugs. And third more important factor is host factors. If humans are infected, so we are using these chemotherapeutic agent against the humans. What is now the host defense mechanism? जो हमारा defense mechanism है वो क्या रहेगा? What is our pathological conditions, or what are our genetic conditions? At what age we are using these chemotherapeutic agents? And most importantly, for activity, to check the activity of these chemotherapeutic agents, is its physiological pH. At what pH these chemotherapeutic agents are using. So this completes your chemotherapeutic agents.